Hello and welcome to our Ensemble Release E75 webinar. So just before we get started, I just wanted to go over some quick logistics. So I'll mute all the microphones, take questions at the end. My colleague Emily is going to be on the chat box. So if you have any questions during the talk, do feel free to ask there. And if you experience a lag in the slide changing, the PDF is uploaded in the materials section of this webinar. So welcome again. I see we have 21 registrants, so that's great. We've got a new release that happened 2 weeks ago, and the highlights of this new release are the new VEP tool interface, which I'll explain what the VEP is, the age of base track, Exxon IDs in location view, GENCODE Basic set, and some more updates which we haven't highlighted just because they tend to be updates that we have every release or every few releases. So talking about what is the VEP? So you've got the URL there if you would like to look into it a little bit further and maybe look at the tool. Basically, you can enter your variants into this tool. So if you have, for example, chromosomal position and different nucleotides or alleles at different positions, you can enter that in. So we take SNPs, insertions, deletions, duplications, and you can find out, is there a known variant at that position, for example, from dbSNP, and RSID, for example, is there an effect on the protein sequence? So even if it is an unknown SNP or insertion or deletion, we'll let you know if the protein sequence is changed at all. We'll also let you know if we have annotated a regulatory sequence there, so perhaps it falls outside the coding region, we'll let you know if there's a regulatory sequence there. And we have different ways to access this. So we've got the web interface. You don't have to know any programming to use that. We have the Perl scripts if you want to use the Perl API, and the REST API as well. It gives you access to the VEP. So what's new then is mainly the web interface. So we have a more user-friendly input form. So for example, this is just the Ensemble default where you've got the chromosome position, the chromosome, and then the start and end base pair, and then the allele there, and the plus is the forward strand. We also accept VCF files, pileup, also variant IDs. So for example, the dbSNP RS123, for example, RSIDs, HGVS notation as well, and you don't have to paste it in. You can upload a file as well if you've got a large file. And our ticket tracker, which is this next image here, is new. Basically, you've got a ticket ID. It will let you know when the results are done and when you can view it. And if you register, which is free, you can do that on our website, you can save these tickets for as long as you want to and go back and view them. So the results are also a new format. So we've got a table with sortable columns, so you can sort by any of these fields. You can customize the table, show high columns, you can export it, and we show you things like, what the consequence is it upstream of a gene, is it missense variant, so has it changed the protein sequence, what is the position in the coding sequence, what is the position in the protein, and we give you hyperlinks back to the gene or transcript or a protein, for example, that it lands in. And we also have pie charts which are showing you the variance according to the consequence or the location with respect to a gene. So you can have a quick view of how many of these are in the coding sequence, how many of these are in, for example, a regulatory region. Okay, the VEP gives access to all our ensemble species, and in this latest release, we have over 70 species. You can see the new species are indicated, new species in 2013 are indicated by boxes, red and green boxes. And if we have a new gene build or a new gene set on a new assembly, so the underlying genome has changed, we show you in a blue box. So it lets you know, kind of, we're getting lots of new species and new assemblies all the time. And so you can just have a look at the ensemble species there. But basically, the VEP, it doesn't require us to have variation information. So even for a species which doesn't have any variant information, you can still use the VEP to analyze your specific alleles and variants. You can also use the VEP on our sister site. So if you're working on plants, bacteria, protists, fungi, or metazoa, metazoa which are not vertebrates. You can go to ensemblegenomes.org. You can use the VEP there as well. So that's all I was going to say about the VEP. I also wanted to talk about age of base. So this is a human-specific track, and basically it tells us when did the nucleotide sequence diverge. So it's coming from the whole genome alignments across multiple species. So if we look at one position in the genome, we can have a look at, is that nucleotide the same across all these different species, or is it different? And if it is different in human, when did that difference happen? So we have a color coding of this track. We've got gray, which means the change happened before the primate branch. Blue, it's a primate-specific change. Red, it's a human-specific change. We only see this difference, or this nucleotide, in human, and it's a fixed, not really variant, so it's a fixed nucleotide. And yellow it's a known polymorphism. So yeah, it's human-specific, it's also a polymorphism. So the track is there. You can see it in region and detail or location view. You can go to the link at the bottom of this slide if you want to actually see what I'm seeing on my screen. And I'm comparing it right now to two different transcripts for BRCA2. So both of those are protein coding transcripts. The golden transcripts are our highest quality set, so we're most sure about those transcripts. And I've zoomed right in to an exon. So we have the same exon in both these transcripts. And yeah, so that might be interesting in the next slide, because now if you click on an exon, you will see the exon ID. So this is anywhere in the site for all species. We used to show you just the transcript ID, the gene ID, and you can jump to the gene, more information about the transcript that way. Now we show you the exon ID. So if you are looking at a particular exon and you want to know the sequence of that exon, you know where to click now. Okay, and there's the share icon, share URL as well. The last kind of thing I wanted to highlight is the GENCODE Basic set. So many of you will know what GENCODE is. Some of you won't, so that's fine. But basically, GENCODE is for human and mouse, and it's a gene set used by lots of bioinformatic projects like ENCODE, 1,000 Genomes. We're using it, Ensemble is using it. Also, UCSC allows you to turn on the GENCODE set. It's the Ensemble default track for genes for human and mouse, and actually it is made from genes that come through the Ensemble automatic pipeline. Just to remind you, those are not ab initio genes. That's coming from cDNA sequences and protein sequences that come from experimental evidence submitted to public databases, and we get them, Ensemble gets them from places like UniProt, NCBI RefSeq, ENA, European Nucleotide Archive. We align all those to the genome and we determine the transcript set that way. Then we have, luckily, Havana, which is a project here at the Wellcome Trust Genome Campus. So they're in the Sanger. And they actually do manual annotation. So looking at genes on a case-by-case basis, looking back at the EST evidence, literature, and coming up with a transcript set that way. So we merge these two together. If Ensemble and Havana got exactly the same transcript, you will see a gold transcript. And that's why I mentioned before that our gold ones are highest quality, because they reflect the best evidence out there in life science databases. So anyway, the GENCODE genes are Ensemble, Havana, and then Yale pseudogenes as well are in there. So the GENCODE Basic is a subset of transcripts. We prefer, in the GENCODE Basic set, we have full-length transcripts for genes where it's possible. So if there's a choice between full-length transcripts and maybe some that look like fragments or missing a start or a stop codon, they won't be in the GENCODE Basic set unless those are the only possible transcripts for a gene, unless there are no full-length transcripts for a gene. So the GENCODE Basic set is there for human and mouse. And I do like to remind people, now we do get a lot of requests from users, can we have one transcript per gene? So we always tell people, well, biologically, that's not always the best answer. There isn't always one transcript that you can choose to represent a gene. Maybe it's perfectly biologically relevant to have 3 or 4 more splice variants. So we don't want to choose just one transcript, but we do want to give you an option to have a subset for the highest sort of quality transcripts. These are the ones we're most sure of. Finally, we've got RNA-seq data for mouse. So we bring the RNA-seq data together to make gene models. And you can turn these on, again, in region in detail or location view, which is the page I've been talking about. And if I turn on the gene models, what I see is these blue transcript structures, and I can compare them to our main gene set. So for example, I've got here several transcripts, so 4 different splice variants for this LUZP1 gene. And then we've made gene models or transcript models off RNA-seq evidence alone, and I've got mouse heart RNA-seq models turned on, and also mouse brain RNA-seq models turned on. And the transcript that is coming up with in the brain, well, it looks a lot like this transcript in our main set. And then the transcript is coming up with mouse heart. It looks like this transcript over here. And just to remind people, if you don't know, our transcript structure is such that exons are boxes. These are exons. Lines connecting them are introns. Filled exons are going to be coding, sequence coding exons. So you can have a look at the transcript structure there. And the blue bar that you see below is actually the genome itself. So this is a way where you can do kind of a quick comparison to see one piece of evidence in the RNA-seq models that actually it is expressed, and maybe it's expressed in this particular tissue. So that's a new thing for mouse. Previously, we only had it for human. Now we have it for human and mouse. And then kind of our more typical updates in a way. We've got a gene set update for zebrafish. So now Havana manual annotation is included. It's updated. So oftentimes Havana will come up with updates to the manual annotation for the human, mouse, zebrafish, or pig, and so you'll often see in our release news that we've updated that annotation for a particular species. DB-SNP build 139 variants have been added to dog, horse, opossum, platypus, and zebrafinch, and actually they're new. It's new for turkey. We didn't have variants for turkey before, so we've added them to turkey, which is exciting, and you can read more there on our news page if you want to know more updates. Next webinar, we're expecting the next release webinar in when GRCH38 is in our main site, which is going to be in the summer. There's going to be lots of new annotation there. That's the new human assembly, because everything has to be kind of remapped onto that new genome. Before the summer release, GRCH38 will be available on pre.ensemble.org, and that will happen in 1 to 2 weeks. And so if you keep an eye on our blog, you can get more news about, if we're doing other webinars, we'll probably will, and then, when things are on pre, etc. And contact us if you have any questions or comments. Just contact us at our help desk. So that's the help desk. We also have a YouTube channel. We've got mailing lists as well. Dev is really good for API users. Courses online. Those are browser courses and an API course, which we filmed, and tutorials page as well. And you can follow us on Facebook. Our Twitter is very active, and we love hearing from users on Twitter. And you can visit our blog as well, ensemble.info. So just to acknowledge the team, that's the Ensemble team there under Paul Flichek. And we've got a load of people to thank for the funding, because clearly without our funders, we wouldn't be able to do any of this. So yeah, we thank them. And then, yeah, any questions, raise your hands.